bioinformatics. So we'll just proceed towards gaining the knowledge with respect to what is structured bioinformatics. Okay, so before going towards the structured bioinformatics into the broad introduction and what are the practical applications, we will just have to know that actually what is bioinformatics first. Then only we can know and we can apply our knowledge with respect to what is structural bioinformatics. So with respect to the topic, you can just imagine that what is structural bioinformatics. Okay, so structural bioinformatics is nothing but we are going to have a broad focus with respect to the uh, 3D structure of the protein or the DNA, and moving towards what are the applications and how actually you can utilize in the field of bioinformatics. That is the main thing. That is the structure of the any protein DNA, and applying the knowledge and moving ahead with respect to studying the particular data with respect to the protein or the DNA, and applying the knowledge and gaining from the different bioinformatics platform, that is the structural bioinformatics. So I think first I should have to take what is bioinformatics first. Okay, then only we can understand that is what is structural bioinformatics. Okay, so the structural bioinformatics, just moving ahead, first we all should know, because many of the participants are from the biotechnology background and some are from the pharma background, whereas the students which is from the bioinformatics background, they have a small knowledge that what is actually the bioinformatics it is. So you can just see the, or you can just split the particular name, that is bio and informatics. So it is nothing but having the knowledge of any biological data. You can just apply the particular biology data with respect to informatics approach. So that is the you can say a simple definition of bioinformatics. Okay, if you have any biological knowledge and that knowledge you can apply so that you can just visualize the data or integrate the data or even to analyze the data using different informatics approach which is there in the bioinformatics field. You can say databases, tools. Okay, so this is nothing but that's a bioinformatics. So it is a discipline, and the quantity discipline where quantitative analysis of any information which has been related to the biology micromolecules you can use, okay, in the form of aid of computer, you can say. So you can use different bioinformatics or the in silico based approach. If you're having any of the quantitative data and you want to just analyze the data further, you can just utilize the particular field, that is bioinformatics. So bioinformatics, it involves the technology that can use in the form of the computer to store, even you can store your data in the bioinformatics platform. Okay, you can retrieve the data. If you doesn't have data, you just have a basic knowledge of any of the disease. For example, you have a knowledge about the cancer disease. Okay, and you want to just want to analyze those cancer data sets, or any of the you want to just interpret those data. So you can take a help, or you can just approach, that is the field of bioinformatics. You can take an your application of that. If you have any you can say that is the data with you, you can just proceed towards the bioinformatics field and take the different databases and tools, and you can analyze your data. You can just visualize your data itself in the single approach. For example, taking the approach of bioinformatics. So it distribute the I have said you that if you have the biological data, you can just visualize the data. Okay, so it has been you can also store your data, you can also retrieve, you can manipulate the data. Manipulate in the sense you can just move your data, that what actually you want for your interest, what particular target you are interested for. So you can be focused on that. You can even distribute and the informations which have been related with the biology macromolecules, such as DNA, RNA, and protein. So this is the overall thing that with respect to the biological data. Biological data is nothing but you are dealing with respect to DNA based study, RNA based analysis and study, and the protein based analysis and study. So you are just focusing on this particular three broad concept, you can say, that is the DNA, RNA, and protein. And taking the help of bioinformatics field, and moving towards the applications of with respect to data analysis, interpretations, and also visualization based. Okay, so that's the field of bioinformatics is. So bioinformatics is limited to the structure, sequences, and the function of analysis of the gene and genomes. So it has been corresponding product that has been offered by the computational molecular biology. So whereas if you know what is the DNA, RNA, and protein, you can just move ahead with respect to the sequence-based analysis, the structure-based analysis, and the structure and also the functional analysis of that. Okay, means you know that what is the sequence, you can just move ahead knowing the what are the functional information it is there in the particular sequence. And after knowing what is the protein sequence or the DNA sequence, you can just move towards going towards the what are the structural changes or the different mutations and analysis are there. So that's the overall thing, and that's the step-by-step informations of that. So this particular bioinformatics have a little history. You can say just go towards that so that you can just be aware about that what actually the bioinformatics is, and for the structural bioinformatics, what particular databases and tools are actually available. Okay, so very broadly, the structural bioinformatics has very broad applications with respect to drug designing concept. Okay, for that, bioinformatics has a very nice different platforms or the databases you can say, where the researchers are trying to utilize the particular platform. That is one of the platform, it is the protein data bank. Okay, so here you will get the protein structures with respect to any of the disease which you have been interested for. Okay, so you should have to be knowledge about that, some history about what is how the protein data bank and how when it is been established. Okay, and also we have the Atlas of Proteins Sequence and Structure database. So these are the number of databases where actually where you all are going to be concentrate, and you will just have to be get the knowledge from there with respect to the protein sequence and the structure. And after knowing what is the protein sequence and the structures, you can move broadly with respect to the protein data bank, where actually they are providing a very broad informations with respect to structural information of any protein or the DNA. Okay, so the major application of the protein data bank is you will get this 3D structure of the protein and the DNA with respect to the X-ray crystallography and NMR spectroscopy based experimental data. So that particular data has been deposited in the PDB data bank. So all the researchers with respect to the molecular biology or from the pharma or from the biotechnology also, they are just moving ahead to utilize the protein data bank for designing a novel drug target with respect to any of the diseases. Okay, so this is the little bit history, and you can also have to know that when the first sequence alignment algorithm has been developed. So it has been developed with respect to Niederman and Wunsch in 1970. Okay, so I think from the students, from the participants which has been from the bioinformatics background, they will have a little knowledge about this particular Niederman and Wunsch algorithm, what we are actually using in the BLAST algorithm. Okay, so here we have the you all should also have to know that when the particular protein first protein structure algorithm has been predicted. Okay, from who has been predicted. So that should you also have to know, and you should be have aware. Okay, so we'll just proceed. That is the particular bioinformatics. So bioinformatics has a broad we can say as application with respect to the databases and the computational biology approach. So what is the databases? Databases is nothing but it will provide you the data with respect to whatever the target or the information you want. That is, if you're interested with the sequence based analysis, the bioinformatics will provide you the different sequence related databases. If you are interested with respect to structural information and analysis of data, the bioinformatics also provide you different structural databases with respect to the particular field of structural bioinformatics based approach. If you're interested with respect to genome and DNA chip analysis or the proteomics or the interactomics based analysis, even the bioinformatics are providing you the different databases and resources in the single platform, that is the bioinformatics. Okay, so why actually we are actually using this, the why the particular databases has been created, because we can curate the data, we can also design our own data sets, and we can also use for data mining concepts. So what are the tools that is broadly there are in, with respect to data analysis is with respect to that is a sequence based analysis. Okay, once you are aware about what is the sequence, you can just move ahead for going towards what is the structural prediction of that. Okay, that is a structural prediction. And once you have a basic knowledge about what is the sequence based analysis and the structural based prediction, you can take your knowledge by applying with respect to docking study, that is the molecular docking study. Okay, so molecular docking study will provide you the 3D structure of the protein, that is you can get from the PDB database, that is a protein data bank, I have already told you. And from that particular thing, I think the whoever participants right now, if they are working in the field of molecular docking study, they will have knowledge about how actually we can utilize the structural information of the particular protein with respect to doing the docking study. Okay, so once you have the broad overview or the knowledge with respect to sequence analysis, structural prediction of the protein, and also the docking study, you can just move ahead your knowledge with respect to the structural genomics based approach, where you will gain the knowledge with respect to the mutations based analysis and what are the structural changes in that particular gene. Okay, and also the functional genomics. Functional genomics will provide you what are the functional based analysis of that particular gene. Okay, with respect to genomics based analysis. And proteomics will provide you the protein information with respect to what are the functional information, what are the families, what are the uh, domains, motifs. Okay, so what are the interaction with that, that is a protein protein interaction. Okay, what is the clinical data which is relating with respect to the proteomics data sets. So it is all integrated, you can say. You should have to be start from one, you should have to be start from one of the particular resource and just integrate your knowledge with respect to by taking the next step and taking the particular different resource, so that you can just integrate your data and analysis and move ahead for designing a very nice application oriented application with respect to whatever the study you have been conducting with respect to any of the disease which you have been interested for. But the flow of the work should be like this in the same manner. Okay, should have to be start from the any of the target disease and just move towards what is the sequence analysis and structural predictions. You can also move for the docking study. Okay, and thus you can move for the interactomics based study and the proteomics and the functionalomics based study. Okay, so this is the particular in silico biology is nothing but the what actually the particular data sets and the information you can actually you are actually working with respect to the field of bioinformatics. So I actually have told you the bioinformatics, it is an integrated field. You can also apply your structural informatics knowledge with respect to sequence analysis. Okay, so if you have the both the information, you can even move for the system biology approach. System biology approach is nothing but where you will just gaining the knowledge with respect to designing the networks or the pathways with respect to protein and the disease. And even you can design the network with respect to protein protein interaction. So string database is there where you can get the protein protein interaction. Okay, you will also get the drug gene interaction database data, so modeling analysis reconstructions, so all will come in the field of the structural biology or the system biology, you can say. And you can even integrate those things. And how, from where actually you will get this particular data, you will get the data from the bioinformatics databases, software, and different tools which are actually available. So you can just utilize the field of bioinformatics with respect to any of the particular disease or the particular topic which you have been interested for. Okay, so our goal of this today's session is to take you towards the ultimate goal of bioinformatics, so that you can just understand the better understand what is the biology or the living cell, and how actually the its the function and the molecular level works. Okay, So you can also move towards what is the analyzing your data with respect to if you have a raw molecular sequence, okay, and you have told you the particular steps. You can just if you have a little bit knowledge about what is the sequence, you can also move towards the what are the structural changes, okay, by taking the help of the field that is bioinformatics, and you can definitely generate new insights or a very potential insights for your particular research, so that you can provide a goal with the perspective of the particular cell or the any of the particular disease which you have been interested for. Okay, so by analyzing the sequence data, you can even use the different informations by generating the if you're interested with the genetic informations, you can also take study the central dogma of biology, okay, with respect to RNA based analysis, and you can also have moved towards the translate of the protein. So this is nothing but the central dogma, okay. DNA you will transcript to RNA, and when it is transcript to RNA, it will move for the translations to form of the protein, okay. So that's the thing you should have to be know, as I have told you, that is the integrated approach. You should have to be moved in the form of sequence to structure based analysis, and once you are aware about the structure based analysis, you can just move towards the omics based analysis, okay, in the form of system biology, okay. So that thing you can do. So the main topics which we are going to cover is what is the structural bioinformatics, what are the different databases are there with respect to structural bioinformatics, okay, and after knowing what is the structural bioinformatics, you can just move towards what are the different databases where actually you will get the data which are related towards the structural bioinformatics, and how actually you can download the data. You have actually you can visualize those data with respect to the protein molecules, okay. And next you can just move towards what is the protein structure, okay. You should have to know that what is the protein structure. You will just definitely get to know what is the protein structures when you will have move towards the what is the PDB database, that is a protein database, okay, and what are the different levels of protein structures, okay, and what are the different representations. So it is very much important, okay, the representation of protein and how actually you can visualize the protein molecule. So what are the broad applications with respect to the structural bioinformatics, okay. This is the broad topics which actually we are going to cover. So we'll just move ahead. So the for respect to the topic, I have told you what we'll cover. Even the with respect to the practical applications, okay. So practical applications is nothing but the after gaining the knowledge about what is this structural bioinformatics, you can apply and you can utilize the particular resources from where actually you will get the data with respect to the structural informatics or the structural bioinformatics based analysis. So UniProt and the PIR is definitely used to understand what is the sequence, okay, what are the informations with respect to the protein sequence. So this UniProt and PIR will provide you the sequence information only with respect to the protein data, okay. So once you will know that what is the UniProt and PIR for the sequence based analysis, you can just move towards the protein structure based analysis, okay. And in the particular knowing what is the sequence information, you can just move ahead for the classification of the particular protein. So classification is nothing but you will get the particular uh, domain, the motifs, the family of the protein, okay. So that information you will get from the databases which is available in the bioinformatics platform, that is the IntPro, PFAM, CAT database, and the PDB database, and the PDB e-fold will provide you the 3D structure of the particular protein, whereas this will provide you that is interpro and PFAM, there is a protein informations or the protein family with respect to the classification. And the protein structure predictions you will also get if the structure is this particular PDB database, CAT and PDB e-fold will provide you the 3D structure of protein which is actually already been deposited in the particular platform. Whereas if you have your own sequence with you, okay, and if you want to just build the 3D structure, You can even build the 3D structure by using the FIRE2 or the Swiss model server. So the particular protein structure prediction will provide you the generating the 3D structure of your own sequence which with you have with you. Okay, and once you have aware about what is the sequence, what is the functional and the classification importance, and what are the 3D structure of that, you can utilize in the form of drug discovery based approach. So for drug discovery, we have one of the database, that is the EMBL database. That is, you will get the chemical informations with respect to the drugs and their drug targets with respect to any of the diseases what you have been interested. So it has been integrated from the EMBL database. So it is called as a CHEMBL database. Okay. So the scope of bioinformatics have broadly, that is, they have been developed for the computational tools and the databases. Okay, so it is why actually it is being used for the gaining the knowledge with respect to the biological concepts, and you can also develop the tools. Okay, and even you can write the different number of softwares by utilizing the sequence structure and functional analysis of the different proteins and the sequences which are there. So these tools are actually used in the area of the genomics and molecular biology research, where actually you can just move ahead for the doing the molecular sequence analysis, molecular structural based analysis, and what are the functional based analysis. I have told you previously this thing. So this particular discussion will move with respect to what are the different biological databases with respect to the structural informatics. Okay, what are the multiple sequence alignment. Okay, because when you have to know that what is the particular biological database, you can just move ahead for doing the multiple sequence alignment. I think from the participants which are from the bioinformatics background, they actually they know that is a blast is one algorithm. There it will provide you the multiple sequence alignment of the protein or the DNA. Okay, or even the RNA sequences. So we have a blast algorithm or the blast platform, where that is blast is nothing but the basic local alignment search tool. Okay, so it will give you the alignment with respect to the sequence, or actually what you have been inputting in that. So you will also get the what is the protein motifs and domain predictions, that is the classification of the protein, and there is a structure predictions by using different tools and databases. And also the molecular phylogenetics is nothing but to getting the classification based on the phylogenetic tree based analysis. There you will get the clades and dendrograms of that, and structural bioinformatics. Okay, and the gene and genomics, I have told you that after gaining the different knowledge, you can just integrate with respect to genomics based approach. So structural bioinformatics is nothing but a discipline from the field of bioinformatics that can grow up with the task and planning with the stream of sequence and the pharmaceutical based goal approach. Okay, so it is an art that can which will give a best results that can be achieved with respect to experts with have a knowledge with respect to, that is the gaining the knowledge with respect to data mining and the tools which are actually there in the field of bioinformatics. So structural bioinformatics, it is related with respect to the, I have told you that first you have to just analyze the sequence, then you have to move towards the structure prediction. So it is really which is related towards the analysis and the prediction of the three-dimensional structure of biological macromolecules such as protein, RNA, and DNA. So in the first slide, I have told you that we are dealing with the biological problems or the macromolecules is nothing but we are dealing with the protein analysis, RNA analysis, and DNA analysis. By after knowing what is the sequence information and just then moving towards the what is the structural information, the prediction of that. Okay, so it deals with the generations about the micromolecules of 3D structures such as a comprise overall folds and local motifs. So these are nothing but the classification of the proteins. Okay, so the principle of molecular folding and evolutions and the binding interactions. So this binding interactions is nothing but you will get the application with respect to drug discovery. So I think the most or the broad application with respect to the structure bioinformatics approach is to utilize the particular knowledge in the field of drug discovery. Okay. Um, and also finding the structure and prediction functional relationship of that particular protein, DNA or RNA. So this is nothing but what actually it is. So in the protein structure, you will have a primary structure, secondary structure, tertiary structure, and a quaternary structure. So primary structure is nothing but the a sequence, okay, of the protein with respect to 20 amino acids. And in that particular 20 amino acids, you will get to know what is the particular mutations being occurs in the single nucleotide, okay, or the gain of mutation or the loss or the missense mutations. That particular sequence analysis you will get to know. Okay, once you will know what is the sequence which is there in the FASTA format, okay, in the gene bank database in the NCBI, then you will just move towards the structure, that is the secondary structure classification and prediction. In the secondary structure, what actually you will get? You will get the alpha helix, beta plates, and beta turns. Okay, so there you will get the particular thing in the secondary structure where the particular, and then after knowing what is here, you will get to know what is the particular. In secondary structure, you will get to know the protein classification, uh, protein family, and the protein informations with respect to using different databases and tools which are there in the bioinformatics platform. Then you can just move towards the tertiary structure of that particular protein, and the quaternary structure will be say used in the form of the doing the drug discovery based analysis by applying the 3D structure of the protein that has been there in the PDB database. Okay, so this is nothing but how it looks like. So if we will if we are dealing with the protein sequence, so we will have the 20 amino acids with respect to the protein, for example, glycine, phenolene, glutamate. Okay, so these are arginine, asparagine, cysteine, leucine, isoleucine, tryptophan, proline, serine. So this all are the 20 amino acids which are there. So it will have based on the particular sequence based analysis and having the knowledge of with respect to classification and protein based on the motifs and the uh, phylogenetic tree based analysis, you can just move towards the structural classification. So these are nothing but how the 3D structure of protein will look like in the PDB database. So if you all are interested to get to know what is the 3D structure, you can just visit the PDB database and get to know how the particular protein 3D structure will look like in the bioinformatics platform, that is the PDB database. Okay, so this particular 3D structure data has been actually been generated with respect to the X-ray crystallography and the NMR spectroscopy based experimental data. Okay, so this is how the classification of the protein begins. So classifications will be done with respect to three broad classification. That is the classification based on structure. So in that particular thing, what you will get to know the is the particular is the fibrous protein or the globular protein or the intermediated proteins. So this particular classification you will get to know broadly once you will be aware about what are the different databases which are there in the bioinformatics platform. Okay, I have told you that is a PIR database or the PFAM database or the ProSite is a database where you will actually get to know the functional analysis and the classification of the protein. Okay, so with respect to the structural based classification, this is the thing what you are dealing. And with respect to the protein classification, with respect to that is a composition, you will get to know the simple protein information and what are the conjugated proteins, okay, in that is alpha helix, beta sheets, and how many chains are there. So that thing you will get to know. Whereas the classification based on the functional analysis is very important, because if you are working with respect to any of the drug designing based concept with respect to any of the disease, for example cancer related disease or the viruses related finding the druggable target for that, so you should have to know the classification based on the functional analysis. So in that it will covers the structural protein, what are the enzymes and hormones informations with respect to the protein, what are the pigments and the transfers of proteins. Okay, So the storage of proteins, what are the toxins information. So this is how the classification of protein based on the structure analysis will carried out. So this is how it works. So if you I have told you that if you have a primary knowledge about what is the particular sequence it is, with respect to here I'm talking about with respect to only protein sequence. So it is showing as amino acid sequence. So it is when it is amino acid sequence in the particular database, so it is called as a primary structure. Okay, so it is in the string form. Okay, so it is in the form primary structure, and if you move ahead to get to know the classifications of the particular protein I have told you, and get to know what are how many alpha helix beta sheets are there in the particular protein, so that particular analysis and classification is known as the secondary structure analysis. Okay, and in the tertiary structure what will happens, the beta sheets and alpha helix will uh, uh, integrate, and it will forms the 3D structure of that particular protein. Okay, based on X-ray crystallography and NMR spectrometry data. Okay, so you will get to know and then you can just apply the particular uh, structural informations with respect to drug designing concepts. Okay, for designing and finding the related drug targets for the particular proteins or any of the diseases which you have been interested for. So we have the biological databases. So what is actually the biological database is nothing but the a database where you will get the biological information. Okay, so how it has been done. So a database which is being comprised with storage and organized of the data in a such a way that if a beginner or if a new users are using or utilizing the particular database, so he will that particular user will get the data, and they can be utilized so that they can actually understand that what particular data they have been deposited based on the search. Okay, so it is very well manner they have been stored the data, they have been organized in a such a way that the informations can be retrieved easily and the variety of different search criteria. So you can even give the search criteria based on the gene search or based on this sequence search. Okay, based on the mutation search if you are working with respect to mutation analysis. So the bioinformatics platform has a broad applications and the databases which are there. So based on whatever the search you are providing, that particular analysis it will start to give you in the single platform. Okay, so depending upon the what data structures, okay, type of the data structure, that the particular database will manage the system, and it will classify based on two types, that is the relation database management system and object oriented base management system. So relation database management system and object oriented based database management system is nothing but if you are new towards any of the database, you can just proceed towards getting the particular knowledge about just searching from the scratch and getting the knowledge about what the particular database is providing to you. Object oriented is nothing but if you are aware about what particular search you are interested. For example, you are interested in T53 gene with respect to cancer database. So you can just start your search by giving the particular search of the keyword, okay, and just move ahead by getting the knowledge of which particular database is providing what type of knowledge or the data for you. Okay, for example, T53 is providing you the sequence, it is also providing you the structural information, the 3D structure, the pathways in that, even the drug targets and which particular disease it is being involved for. So this is how the particular different databases it work. Okay, so all the bioinformatics databases and the platform it will work in the integrated based approach. So whenever you are new and you are finding any of the one database, it will link you with respect to another database in the single platform, so that you can just view the different data how they have been designed and they have been structured, and you can utilize those things. So based on the biological databases, the bioinformatics have been broadly divided into the three category. That is the primary database, secondary database, and the specialized database. So this is how it works. So if you are new and you are beginner, you can utilize the primary database. In that, you will get to know different sequence data. Okay, and in that some classification of the data. In the secondary database, you will get to know the 3D structure of the particular protein or the classifications or the particular different drug targets and the pathways. And in the specialized based database, you will get to know which particular information you want with respect to only pathway or only omics based approach or the NGS based analysis. So this is how it works. Okay. So this is nothing but the databases which are there in the bioinformatics platform, Uniprot, PFAM, okay, CAT database, SCOP database, Panther database. So these are all specialized database, genomics database, okay, ProSite database, PDB database or the PDBJ. So NCBI PubMed database will give you the literature database, okay, with respect to literature of the biomedical database and analysis. Keck database will provide you the information with respect to pathways. Okay, this is how the multiple sequence alignment you can also do by using the cluster W. So these are the number of pathway number of databases which are available in the bioinformatics platform. So the structural bioinformatics, which also refers as analysis of the micromolecules of structure, right? So you are dealing with the micromolecule analysis. After knowing what is the sequence analysis, then you can just move towards particular with respect to the protein, and you can utilize the different platforms and tools which are there in the bioinformatics platform. So this process will use to predict the 3D structure. Okay, structure bioinformatics is nothing but you are dealing with the structure analysis and prediction of the data. So you will get to know the 3D structure of the particular protein, okay, with respect to the analysis, and you can compare the sequence and alignment, okay, the secondary and the 3D structure predictions. So this is actually the broadly how the structure bioinformatics works. You have to just focus toward the 3D structure, and that you can also view the sequence alignment and what are the secondary and 3D structure prediction tools and databases are there, okay, and how you can also use in the form of structural biology based analysis. So these are the actually the techniques or the particular uh, databases and tools you can say in the field of structural bioinformatics. Okay, so PDB, Scope, CAT is the databases. If you are interested for the molecular visualizations, you can use the RAS small Swiss PDB viewer. So these are the different databases and tools out there. Protein structure prediction. So I have told you the protein structure predictions are in the form of secondary structure prediction and the tertiary structure prediction. So in the secondary structure prediction, the FIRE or the particular different databases are there. And the tertiary structure prediction you can do by Swiss model server or the modeler, and also you can do the homology modeling. Hmm. And if you are also know that what are the protein structure and the 3D structure databases are there, you can also look towards the structural analysis or with respect to protein-protein interaction prediction. So I have told you the string database is also there. It will provide you this interaction-based study. And you will also get to know what is the protein structure comparison, okay, and protein structure classification and the molecular docking study. So these are the different tools and databases are there. So for structural bioinformatics, the databases which have been used and they have been stored with respect to the PDB database that is based on NMR and the extracrystallography, okay, and that is the PDB database you will find in this particular link. And we also have the PDB sum database and SCOP database, and also the CAT database is also available. You will get the data from here. So hey, this is how the X-ray crystallography experiment is this. So this is the instrument where the particular 3D structure will predict and based on the sequence, and it will get deposited in the PDB database. So this is how the particular 3D structure of the particular protein, it will start to be generate based on the X, Y, and Z coordinates of the any of the molecules. Okay. So as a PDB database, it is basically um, so PDB database is broadly a database which is related with respect to structural bioinformatics. So it is updated weekly. Okay. So it has a certain, so I've told you that it is predicted based on X-ray, NMR, and even the electron microscope data. So what particular structures it predicts? It predicts the protein, nucleic acids, and also you will get the complex structure. Complex structures is nothing but the complex structures of the particular protein which is attached with the particular drug. So that's the complex structure also you will get. So this is how the PDB database it has been structured, and they have been deposited different data. So this is the what. So you will also get the particular a small structure, even the microglobin structure also you can get. You can also get the lysosome, hemoglobin, and many more structures with respect to the 3D based analysis. So this particular 3D structure analysis it's been providing with respect to the PDB database. So structural bioinformatics, why it is very important for the analysis and doing certain applications with respect to bioinformatics or drug discovery based approach, because it is the particular structure bioinformatics is integrated. Because based on the structure analysis, you can also move towards with respect to the sequence based analysis. You can also move for the pharmacokinetic analysis. You can also have a broad application with respect to system, that is a system biology approach. Okay, with respect to biomedical approach is nothing but you can just take any of the target with respect to any of the disease, and you can just move for the system biology based approach, and getting towards what are the biomedical based applications with respect to structure bioinformatics approach. And in the bioinformatics you will have different algorithms, that is I've told you that is a blast algorithm. You can do the sequence alignment. You can move for the structure based analysis with respect to the structural analysis of any protein and DNA. And you can also move for the biological networks and system biology for getting towards what are the databases and the interactions are there. And also you can move for the proteomics based analysis. Okay, so this particular informatics have also a broad application with respect to literature data. You will also get the literature of that particular informations. Okay, so this is how the structure bioinformatics has been integrated. So all the data which has been related with respect to the pharmacoinformatics, biomedical informatics, and even the bioinformatics with respect to the literature or the pharmacological data. Even you can move with respect to drug designing in the form of personalized medicine. So that all are integrated with the structured bioinformatics. So going towards any of the broad applications with respect to analysis the particular data, you should first have to know what is the structural bioinformatics. Okay, so this is how the particular PDB database work. It will have a different structures which has been deposited, and it has been weekly updated. So you can utilize the PDB database to start your particular data analysis with respect to any of diseases. So this is one of the uh, book where you can refer, okay, for doing the particular structure and bioinformatics for the analysis and gain the knowledge by using this particular book. You can take this particular book screenshot and take the structure bioinformatics ebooks are also available. You can utilize that. Okay, so this is the you can say in the single side you will get the application about how the 3D structure is why very much important. Why? Because if you know the structure bioinformatics with respect to 3D structure of any protein, you can move for the analysis based on the interaction based study and getting towards the what are the force and the protein classification based analysis. That thing also you can do by knowing the 3D structure. Okay, you can even study the molecular interactions based on the, for example, molecular docking and the molecular simulation space study. Okay, you can also gain a knowledge with respect to what are the electrostatics property of that particular protein, and what are the protein ligand complex. So this is will required for the drug designing concept. So I have told you the broad application for the 3D structure bioinformatics in the field of drug discovery. Okay, so you can also find what are the residues and what are the conserved regions in the particular protein and 3D structure. What are the mutations in that? And once you know what are the mutations in the particular protein, you can also move for the omics based analysis based study of the system biology approach, or even the omics based approach after knowing what is the structure bioinformatics. So this is how the particular 3D structure of the X-ray crystallography will work. So the first they will give you the sequence, okay, so in the form of the what are the protein folds and the mutations in that. Then based on the particular technique of the purification crystallizations, the particular data will be collected, and it will form a structure of that particular data, okay. And once the particular structure has been formed, it will start to refine the particular structures. And when the particular structure will get refined, you will get to know what are the functional annotations in that particular sequence. And that particular functional annotations and the particular alpha helix
Exactly.
beta sheets, we are getting to know in the particular PDB database. Even you can publish your data with respect to that, and even you can deposit. So this is how the sequence to the structure it will get generated, okay. So the particular protein which will be extracted, and it will based on the particular sequences it will start. So in uniprot we have nearly 45 million protein sequences available in the protein database, that is with respect to that uniprot database. So you will also find the 3D structure of proteins, that is the protein structures, their folds and their superfamilies and families information in the scope database. So this is the applications, or you can say where you actually will get the data after knowing what is the structure of informatics. So this is how the PDB database, the 3D structure it is providing to us with respect to alpha helix, beta sheets, okay, and what are the chains, how many chains are there. Based on that it will provide. So that's the overall thing. So before moving towards the conclusion part, I just want to demonstrate you one of the database, that is the PDB database, so that you will get to know that how actually you can utilize the particular structure bioinformatics applications. So I will just share the screen with you. And what I'm doing, I'm just opening the PDB database. So I will just show you that how uh, the practical demo will work, and how actually you can utilize the system structure bioinformatics with respect to taking one demo of that. So this is the PDB database. Okay, I think more of the students are aware about the PDB database, but this particular PDB database is very interactive and very application and the knowledge based database, because as you have seen that recently the COVID-19 pandemic has been evolved, and every researchers are trying to get a novel druggable target with respect to that particular virus. So this particular PDB database is providing you the drug targets for that particular virus. So here you can see the PDB database, that is a protein data bank, has a this number of, that is the biological macromolecules and structures which has been available with that. Okay, you will get to know the overview of that. Even you can get the resource only with respect to the coronavirus, that is a COVID-19 related drug target. So I have told you that every weekly the PDB database will get update. So now we have the molecule of the month of the December, that is the it has been reflecting over here, that is you will get over here. If you are interested to get more on this, you can just click on that and you will get the more information with that. You will also get a certain publications, okay. You will get to know that the least entry, so least entry is nothing but the how many different structures particular data has been worked. That data you will get to know from here. Whereas here is the particular structures. So PDB glance has the structures with respect to this have actually been deposited in the PDB database, and structures with respect to human sequences and the nucleic acid data. So more statistic also you will get. So I will just search one of the protein for your particular interest. So that, for example, P53 gene is universal. So whenever you will click on P53 gene, it will start to give you number of different related hints. So you can click on one or directly search on the PDB T53, and this is the particular page you will get. So here it will provide you the how many structures are available. Okay, and you here it will show you that how many structures with respect to the organism it is have. There is a homo sapiens, mouse, daniel rio, and many more. So you will also get the taxonomical information. So here is nothing but the experimental methods, that is x-ray, microscopy, and what are the protein related structures and what are the DNA related structures are available. And these are the different enzyme classification. So this is all the 3D structure of the protein. As per I've told you that the protein classifications. So here in the single platform, you are getting all the information. You're getting that from which organism the structure has been deposited, the taxonomical information, the experimental methods. You will get the particular protein informations with respect to is the particular target is protein target or the resolution of that you will get because it has been defined based on the X-ray crystallography and NMR spectroscopy data and the release date. So you will get to know that if you are any of the target you are selecting, so when it has been released, it has been recent data you are working or the updated outdated data. That thing also you will get to know. You will get to know what is the enzyme classifications. So that is the if the particular target you have selected is a transferase, hydrolysis, ligase or the oxidase, isomerase. So like this, you will get the classification of that. You will get to know what the membrane protein name, if it is there, and the symmetric information, asymmetric, cyclic, or dihedral. Okay. So from here, this is nothing but the four-letter code of PDB you will get. Okay, so this particular database, the particular T53 gene, it has been, they have given their own unique identifier in the form of four-letter code. So this particular 3D structure has a 6IUA four-letter code, and it will give you the title of that and when it has been released. So 2019 of the particular data is there, it has been released. The method, that is the X-ray crystallography, and this is nothing but the angstrom, the resolution of that particular 3D structure, and which particular organism they have been studied, and is there any particular micromolecule has been there, for example that the complex structure of that. So that particular data, the overall means the overview you will get, and here you will see that the download file option is there and even the view file option is there. So I will just show you one of the 3D structure. So I will just click this one. So this, by clicking, you will get the broad overview of that. Okay, the four-letter code of the particular PDB structure. You can even download in the PDF format, and even you can take the FASTA sequence. And these are the other zip file format and different file formats which are there. So DOI ID of the publications also been integrated related over here. Okay, so you will get to know the author names and the index mental method. You will get to know which particular method, the resolution, R value, R free value. So these are the different values. Based on that, the 3D structure has been generated. So WWWPDB validations is also been done. So that particular link they have also been given. Okay, literature data they will also give you. If they have the literature, they will give you the abstract over here. So this is nothing but the chain, how many chains are there, the sequence length is been defined based on the protein sequence. So based on that, it was providing you the for example amino acid sequence, how many strings are there. So 275 string length is there. So this is the 3D structure. So I have told you that the bioinformatics database is all integrated. So whenever you are searching with respect to any of the data, the different data will definitely be integrated default. So here you can see in the PDB database, the UniProt database is linked. So here you will get the UniProt ID over here. So once you click the link, you will get to be open in the UniProt database. Okay, even the GTX, that is the genome based analysis of the data, is a GTX is one database. So that particular ID is also been provided. So if you click, you will just reopen in the particular database. So it is all linked. Okay, it is all integrated database. So you will get the reference sequence and of the particular protein. Okay, so that which particular positions of that, and what is the particular position of the particular amino acids. So methionine. So like this, you will get the positions of that. And here you are getting if the particular 3D structure is have some complex. So this is the it have a drug which is been interacting. So this it will show you the structure of the drug. So I think the students from the pharma background, they will have to they will know about how this particular uh, drug structure looks. Okay, so if it is having the 3D structure, it is in the form of complex, so it will give you the drug information. Okay, and in the ligand interaction, it will show you that how particular drug is been interacting with the particular protein. So that particular it will show you the 3D interaction. So this is how it will show. Yes, okay. So this is the particular thing it will show you that the particular how the drug has been bind. So this dotted line is nothing but the active site where the particular drug goes and bind in the particular 3D structure of the protein. So it will provide you the view of that in the form of the, and the red dots are nothing but the water molecules. Okay, so this is how our 3D structure will look. So you can design any drug target for the any of the disease by utilizing the PDB database. Okay, so you can change the different orientations of from here, for example, cartoon. Okay, so here you can do like that. So you can utilize the thing from here. So you will get to know if you are just scrolling around, you will just highlight that what are the uh, amino acids, threonine, and which particular chain it involved. So this is how it will show you. Okay, if I go in the particular site, it will show me that which what is the particular amino acid has been involved. That is histidine in the 16 positions. So here you can see the arginine and 20 positions. So like that, it will show you that which position the particular drug has been interacting. It will also give you the overview of that based on the sequence also. Okay, so this is how the it will show you the 3D structure of the particular protein, and the rest other informations also it will provide. So here from here you can just also download. This is the structure summary for the 3D view. You can just click on the 3D view and see the different view information right now what we saw. And the annotations, it will show you that how what is the mutations and annotations with respect to the here this is nothing but the structure bioinformatics is here is nothing but you will get the domain informations of the particular protein from the cat database. Okay, so here the cat database is linked for the protein family annotation information, you will get to know from the PFAM database. Okay, the gene product information you will get to know by gene database. Okay, so here is the all information has been linked from the protein sequence to protein classification, family information, and the domain information, and also the structure information. In the experimental matter tab, you will get the from which particular experiment the particular structure has been involved. So it is for it involved designed from the 3D structure, that is X-ray crystallography. So likewise, the diffractions, the resolution, the different information you will get over here, and the sequence you will get to know what is the sequence of the particular protein. Okay, in the form of 3D structure also, or in the form of the faster sequence. So what is the active sites of that? So this is all it will provide you. And the genome, it have been related because if you are interested to study the particular protein sequence based on the reference genome analysis, so on which particular chromosome the particular mutations has been occurred. So that thing you can just visualize by using different tabs which has been over here. So that's the major application of the structure bioinformatics by using the PDB database, or else the different database if you want to do the classification and doing the phylogenetic tree analysis. So there are the different databases are available. That's I think the broad overview with respect to the introduction and having one simple demo in the form of practical. That is the just in the form of viewing the particular PDB database and get to know that how actually the structural bioinformatics have applications in the field of the bioinformatics, and how from where actually we can get the data, how we can visualize and how we can analyze your data. So I think that's the is the overall information with respect to the structural bioinformatics, which you have been all enrolled for. If you all are having any query, they can ask me. So I think that's the broad introduction with respect to the structural bioinformatics sessions, which and the practical applications of that. So I hope that in future you will try to join